Our final section in Chapter 6, dealing with microbial growth, deals with how microbes divide, specifically looking at the growth curve. And so one of your objectives here is to be able to list and describe the 4 phases of the microbial growth curve. We're also going to look at some ways of counting microbes. We divide those into direct and indirect methods. So just a little reminder, when prokaryotes reproduce, and here we're talking about bacteria, they use binary fission. And then some microorganisms might use budding with the formation of kinetospores, and then fragmentation of filaments. And so some of these, binary fission is more of the bacteria. Some of these might be like yeast, use budding. And some of these might be some more of your fungi as well. But with binary fission, what the bacterium does is it first of all has to replicate its DNA. Just like a eukaryote, we need to make sure we copy the DNA before we can divide the cell. And so remember that bacterial cells have one like large circular chromosome. And so they have to replicate that first. And then the cell wall and plasma membrane start to pinch in and constrict a little bit there. And then finally a cross wall forms, dividing those into 2 separate bacterium, and they each have their own copy of the DNA in there. So binary, bi means 2, so literally just copies the DNA, splits it into 2. And this just shows you exponentially why we can get so many bacteria in a short amount of time, because of how they can reproduce. Especially some of them can go through binary fission every 20 minutes. And so exponentially you can get quite a few microbes. That's why when we put bacteria in the incubator, the next day you'll see a whole bunch of growth on your plate. That's already, millions of bacterial cells there. And so we can look at your bacterial growth curve both using over generations and looking at numbers of cells, or we can look at the log number of cells as well. And you can see that exponential growth happening like that. So there's 4 main phases of growth that we're going to look at when we look at a microbial growth curve. The first one is called the lag phase. It's basically kind of a flat area right at the beginning of the growth curve. It's flat because at this point the bacteria is just kind of adjusting to their environment, so there's very little growth. They're getting used to the temperature, the nutrients, all of that, so there really isn't a lot of growth. But then it really takes off. We call this the log phase, also known as the exponential growth phase. This is where you'll get the maximum amount of growth occurring. And this will continue as long as cells have the adequate nutrients and favorable environment. So if there are no limiting factors kind of holding them back, those microbes are going to keep reproducing, going through binary fission, and you get more and more and more, and you'll get that exponential growth. But eventually, you're going to reach what we call the stationary phase. That's where the rate of cell growth equals the rate of cell death. And that's because now nutrients are going to start to get depleted, along with oxygen. More and more wastes are going to build up. And so now you're starting to have a few limiting factors stopping the growth of those microbes, so they won't just keep on reproducing. Now the rate at which they're reproducing is equal to the rate at which they're dying. And so you'll get this very flat top part of the growth curve where you won't get any more growth. Eventually, you'll go into what is the death phase, and that's when the limiting factors intensify. More and more waste builds up. There are less and less nutrients available, less and less oxygen available. And so basically now the microbes just start dying exponentially in their own waste. And you'll see a sharp decline in the growth curve again. So here's what it looks like. Here's that lag phase. Microbes are getting used to their environment, so there's really no growth there. They're just kind of hanging out, kind of getting used to it. Then you have this log phase or exponential phase where there's rapid growth because they've got tons of nutrients, lots of space, lots of oxygen, life is good. Then they start to run out of space, nutrients, so on. Waste is starting to build up a little bit. So the amount of growth equals the amount of death. So they just kind of even off up here at this flat part. But then the limiting factors build up. Too much waste, not enough food, not enough oxygen, and boom, they just start to go through this sharp decline here in the death phase. When we count microbes, there's different methods. One of the things we can do, because microbes can reproduce so fast and you can get so many that it'd be really tough to count them all, we do what are called serial dilutions, where we have your original sample here. We take the original sample and we put 1 milliliter in 9 milliliters of just plain broth. So we dilute it 1 to 10. Then we take 1 milliliter of that and put it in 9 milliliters of broth. So now it's diluted 1 to 100. And now we take 1 milliliter of that and we just keep doing that and doing that and doing that. Serial dilutions. We're not talking about Captain Crunch or Cheerios here. Serial means in series. So we keep diluting, 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 diluting. And you can see how it gets lighter and lighter and lighter and lighter. So there should be less and less and less bacteria in there. What we do from there is we plate it out. When we plate it out, then we can actually look at the number of colonies that grow on there and determine how many colonies grew just by doing a count on the plate. And so after incubation, we count colonies on plates that have either between 25 and 250 colonies. So when you look at that first dilution, 1 in 10, oh, it's just a massive growth. That's what some of you guys get when you're inoculating your plates. You can't even see separate colonies. Even with a 1 to 100, still pretty thick. 1 to 1,000, still pretty thick. Oh, now we can start to count. 1 in 10,000. Now we can 1, 2, 3, 4, 5, 6, 7, 8, and so on. So we can look at the number here and count, and then we can do some mathematical calculations to see how many microbes are there that would have been in the original broth sample. Another way we can do it is by membrane filtration. We can actually take the microbes and in the water, and we can have them on a special membrane filter, and we can plate that out, and then you can look at these right here, and you can just start to count all these. Oops, I better use a different one so you can actually see. You can actually count all these different microbes here that were on that special membrane and count them. We can also use what's called a most probable number, and this is for microbes that grow in a liquid medium. And we take that sample, and then we dilute it, and we compare it to a statistics table. And we look at here, if we took 10 milliliters out, then 1 milliliter, and then 0.1 milliliter. How many of these would change to that yellow color? Okay. And so then we look at the number of positive tests here to see. Here we had 5, and then we had 3, 2, 2, and here we had 4 and 1. And so we want to look at that as we compare it to the statistical table, and that kind of helps you estimate the number of bacteria in a sample. They oftentimes will do this from liquid cultures. So like if they wanted to see, every once in a while when you hear people in some of these small towns have to boil their water because they found E. coli. Well, they probably did this test and they found out they had too much E. coli in their water. Using the statistical analysis, they had too high of a E. coli count in their water. And so then they're told, you need to boil your water until we can get this back down to better numbers, acceptable numbers. Another way you can do this is through direct microscope count. There's a special slide called a hemocytometer, and we often use it when we're doing blood counts, like white blood cell counts. And so it's actually a microscope slide that has this little grid on it right here. And so magnified, it looks like this. And we can count, within the grids, how many bacteria we see. Kind of like if you were going to do a white blood cell count. You'd take a blood sample and you count how many white blood cells you see in that grid. That's what they're doing with the bacterium. So that's a direct microscope count using a special slide with grids. That's another way of doing a direct count. So all of these are more direct counts of counting bacteria, where you can actually see the results and actually count colonies or count them on the membrane or look at positive and negative results and do an estimated count using a statistics table or counting them directly on a special slide. Indirect ways of counting microbes, one of them is turbidity. And with turbidity, we take a sample. Obviously, if you've looked at your microbes when you inoculate a broth, if it starts to look cloudy, that means you've got bacteria growing in there. So we use a special piece of equipment called a spectrophotometer, and it transmits a beam of light through your suspension, through your broth, and records the percent transmission, okay? And it also shows the absorbance or the optical density, okay? So if you have no bacteria and it's totally clear here, all the light goes through, it's 0% transmission, okay? I mean it's 100% transmission, but 0% absorbent. So it's able to transmit that light 100% all the way through, okay? But if you have cloudiness, or what we call turbidity, because you have a bunch of suspension that has bacteria, not all of the light can transmit through, okay? So here you can see the percent transmission is only 20%, okay? That's because some of this, as soon as the light hits it, it hits some of those bacteria and it refracts off and bounces off, okay? So not all the light gets transmitted through. So based on those turbidity percentages, we can calculate then how much the bacteria has grown. Some other ways we can look at indirect are, besides turbidity, metabolic activity. That's by looking at, doing some of your tests like fermentation tests. Obviously, if it can change the phenol red tubes from red to yellow, we know there's some activity there. We know that your bacteria is growing and dividing if it can do that metabolic activity. We can also measure the dry weight of the microbes to determine if you have a heavier amount from one period to the other. That means that they've divided and there's more of them. Again, the plate counts, filtration, most probable number, and direct microscopic count are all direct methods. Turbidity, metabolic activity, dry weight are all indirect methods. We're not actually counting the microbes, but looking at other results to kind of let us know how much they've divided. So that's where you measure microbes. And again, make sure you know these phases of growth and what's happening at each stage.